Hello and welcome back. In this video we'll cover two main areas. Very briefly we'll talk about how it's often useful to classify these large numbers of bioinformatics databases that we're being introduced to here in the course, as this will guide our thinking and our prioritization of resources and the types of information that we can get from these different databases. Now, for the lion's share of this video, what I want to actually do is give a little vignette. That's a task-oriented introduction or walkthrough demonstrating how these major bioinformatics databases that we've talked about this far intersect, and how that's a useful thing, how we often find ourselves jumping off between and linking out to other databases because they contain complementary and indeed useful information that will improve our knowledge and our workflow as we go through. Now, this third point here we'll highlight as we go through. We'll talk about some of the major bioinformatics formats. That's how the actual data is stored within these databases. We'll highlight that and kind of learn it on a need-to-know basis as we delve deeper into the course. But I will highlight a couple of those major formats, for example, how sequences are stored and how structures are stored in some of these databases. So first up, all these bioinformatics databases that we've mentioned thus far, they can be usefully classified into so-called primary, secondary, and some people call it tertiary, but most often you'll find it as composite databases. And this is according to the kind of source data that they contain. Now, primary databases, also you'll hear it referred to as archival databases, they consist of data derived primarily from experiments. So the classic examples here are GenBank, that's NCBI's primary nucleotide sequence database, and the PDB, or the protein data bank. This is the major repository or database for biomolecular structures, primarily from X-ray crystallography, but also NMR and cryo-EM. Now, these two primary databases, they contain information from the experiments, whether that's from the crystallographic experiment or from the sequencing experiment. The data in these databases is owned by the experimental person or group that deposited in those databases. Now, they are what's known as redundant because many people are interested in, for example, the same protein for drug development purposes. They'll solve multiple protein structures, or they'll sequence it in multiple different forms, different groups from around the world. So they'll be redundant. There'll be multiple copies of the same thing in there. Also, because it's owned by these experimentalists, if there's a mistake in that data, the database maintainers, they don't actually have the ability to go and correct it. They can flag it and say there's a mistake in it, but they need to go and talk to the original experimentalists and say, could you please fix these entries? And sometimes that doesn't happen. So both of these aspects, but primarily the redundant nature, that there's many copies of the same or nearly the same thing in these databases, kind of motivated the development of these secondary databases. These are also known as derived databases. They contain information derived from the primary databases. So a classic example here would be RefSeq. This is a non-redundant set of created reference sequences, that's where the ref comes from a GenBank in this case. Okay, so GenBank and RefSeq, they're related. RefSeq contains derived information from GenBank. The idea here is that it's non-redundant, so you're going to have the best single copy of a given gene, for example, sequence in RefSeq, whereas you have multiple redundant copies in GenBank. Then finally, we have composite or meta databases. These join a variety of different primary and secondary databases together. They're kind of a one-stop shop for all sorts of information. They pull stuff from wherever they can get it usefully and have consequently lots of useful link outs and resources to jump off to the primary databases and the secondary databases and all the rest. But it's a place where you can kind of catalog all sorts of things together. Now, good examples of composite databases include OMIN. OMIN stands for the Online Mendelian Inheritance in Man. It's unfortunately named with the man component there. Of course, we mean humans. But this is an old database. It actually was originally a set of encyclopedia-like books that was distributed and sent around to primarily clinicians who are interested in the genetic basis of various human diseases. And then, of course, as things developed, it became online, but it kept its name, that OMAN name. Another good example here is the gene databases that we'll use a lot. It's at NCBI. It's a great starting point for exploring, you got it, genes, right? This is molecular data, literature data, transcription data, structural data, everything that they can find for a given gene, all assembled and put together in this gene database. And it has extensive link out, kind of interlinks or URLs out to other databases. So it's a really good place to start investigating, say, a new gene that you maybe start a new research project on, for example. Okay, so let's explore some of these databases. Let's do one of these little vignettes. So let's imagine for a minute, if you bear with me, that you've just come out from a seminar, you've heard an online talk, or you've read a paper, and it was about gastric cancer, let's say. And then one of your colleagues, one of your buddies, or a coworker asks, oh yeah, what do you know about that gene, let's say it's KRAS, that the speaker was talking about? Now, I picked KRAS because you've maybe heard of RAS before in other classes, and we'll maybe be able to bootstrap our knowledge here as we're able to go through these databases. But just substitute in whatever other gene you want for RAS or KRAS in this particular case, and follow along in the next few minutes with what we work through. So how would you find out more about this particular gene? Well, you could just Google it, and you'll get lots and lots of hits, of course. Many of those will be unrelated, right? Some of them may even be misleading and not from scientists at all. You could do, if this wasn't spring quarter of 2020, you could go to the UCSD library, which of course we can't because it's closed. And actually, does anyone go to the library anymore anyway? I'm not so sure our students do that. But what I'm going to suggest that you do is have a much more focused and intentional approach, and that's use these bioinformatics databases at the NCBI and the EBI that we've been learning about thus far. So let's start. We'll go and we'll visit this URL to go to the NCBI database. And what we're going to do here is we're going to type in the gene that we're after. So I'm typing in just RAS up here in this top search box. So this is this top search box here. This is what we call entrees. It's the way to search across all databases. You see that from the drop-down menu beside the red square here. So it's going to search for RAS across all of NCBI. And when we do this and push return, it's going to link out to lots of resources. But first, before we look at those results, let's see what sort of questions we want to answer. So here's the kind of questions that we're going to go after. What chromosome, so what's the genetic location or the genomic location, for example, of these genes? What other genes are in the vicinity of that query gene? This is often useful if you're interested in what comes along with a given gene or what multiple mutations, for example, might affect. And for that, we're going to use NCBI gene. The second question here is, what can you find out about the molecular functions, the biological processes, and maybe where it's located? For that, we're going to go and jump over to Europe and look at this GO database, what called the gene ontology resource. And then the third thing we're going to ask or try and answer here is, what amino acid positions in the protein are responsible for ligand binding? If this is a ligand binding protein coding gene, we're going to use Uniprot, the premier protein database, also over at the EBI. And then the fourth question, what variants of this gene are associated with gastric cancer and other human diseases? We're going to go and visit that unfortunately named NCBI resource called OMAN, Online Mendelian Inheritance in Man, in this case. For our penultimate question, what's known about the protein family, more generally, its species distribution, so where is it in the tree of life, what kind of organisms is it found in, how many copies of it are there in human, how many variants are there, what's the residue-wise conservation? That would look at, again, what are the functional bits, those that are conserved over the course of evolution. We'll turn to this EBI maintained database called Pfan, the protein family database. And finally, then, just like we did in our first hands-on session, are there high-resolution protein structures available to examine maybe finer grained details of these mutations or their functions, and maybe the functional effects of these mutations? We'll turn to the PDB database, the protein data bank in this particular case. So here's the results of searching NCBI at the time of recording. Now your numbers will differ from this, okay? This slide was actually generated for the last quarter's class, and I've put it here, but notice how your numbers differ. These databases are growing tremendously quickly. So see what the delta is. What's the number that you find in the gene database compared to this number of 87,000 entries here. So this is where we're going to go first. I'm going to click on the gene entry, and this will take us to all sorts of information here on things that are found with RAS. Now we're obviously interested in the seminar fictional seminar was about gastric cancer, and we're kind of human-centric, human-biased in that seminar. So we're going to click on the top organisms here. We're going to say Homo sapiens. So I'm going to click on that. That'll filter our results down to humans. And notice what's happened. Up in that entree search box here, I've put a yellow rectangle around this. I did not type this in. It was automatically inserted, if you will, by clicking on that Homo sapiens link. It's written RAS and Homo sapiens with this strange code here, which stands for the taxonomy ID. It's a database of taxonomy, and this is the identifier for humans in that particular database. So the AND bit is the bit that's important here, the AND. And this, of course, is just set logic. If you remember back to those early days at school, the AND means the intersection of these two things, both RAS and disease, for example, or RAS and humans, right? As opposed to the OR, which would be both the union of these things, or the NOT, which you could also use. Now I'm going to go back to that website, and most people, don't write these things in the search box. You can if you want. Most people use this filters. They click on the sidebars, either on the left or the right hand, to filter and search within our results. And I can see the second entry that I've highlighted here is already what we're after. It's KRAS. So I'm going to click on that. It'll bring up the gene entry for KRAS. And the way we navigate this gene entry here is we use this right-hand side table of contents. This is kind of the link out to all the portions of this gene page. If you scroll down, you see that red arrow. This is a really long entry, a single web page that's extremely long. And it has all these subsections. So by clicking on this right-hand side, what you'll do is it'll just scroll you down to the section in question. So our first question is, so what chromosome location and what genes are in the vicinity? So I'm going to click on genomic context here, and that'll bring me down the page, and I'll see that this is on chromosome 12. It's on the P arm here in this case, right? And this refers, this is the old school cytogenetic kind of location. So it's the short arm of chromosome 12 here, and the 12th band from the centromere. So this is based on those distinctive patterns or bands created when chromosomes were stained with certain chemicals. And it describes the location, the particular location here on the chromosome. Now we will also see that it's got 6 exons here. And so how would you know how many introns it has? Well, 5, right? One less than the number of exons. And we can see here also in the red box, this is a little schematic of the chromosome 12 region. This is the second red box. Here we can see genes that are in the vicinity of it. So there's our KRAS gene, that's the red arrow here. And then these other genes are those in the vicinity. And if you were using this in your own computer, you could put your mouse over one of these other arrows and click on it to link out to that particular gene entry to find out more about those neighboring genes on chromosome 12, for example. So on to the next question. What molecular functions or biological processes and cellular component information is available? Now you could just start reading from the start of this page, and it'll be a bit like reading a scientific paper, where you have your abstract or your overview here that'll have some human written verbiage about what KRAS is and what it does, and that's useful. But the big problem here is, humans are humans, and humans are different, and the way I write is different than the way you write, which is different than the way someone in England writes, or someone in India, or someone in China, or someone in Australia. They write in different ways. And sometimes it's hard to know if we're writing about the same things. So because of this, we have a standardized way of describing things like function and cellular location. It's called an ontology. And one of the major ones, one of the ones I want to introduce you to first, is called the gene ontology. So if you see here in the sidebar, there's a link to gene ontology. When I click on that, it'll scroll down here, really down, quite far down the page, and it says gene ontology provided by Goa, G-O-A. This is not that nice area in India where you can go for your vacation and eat lovely fish-based curries and things like that. This stands for the Gene Ontology Association, and it's actually maintained out of the EBI, the European Bioinformatics Institute. So let's, you don't have to do this, but I'm just going to click on this and it'll take me over to the EBI. It's this, it tells you more about this. It's a control vocabulary of terms describing gene product characteristics, such as function. Okay, so let's go back and say for a minute, why do we need these things? What are these ontologies? What are they useful for? Well, we all hopefully can appreciate that knowing what something does is pretty important. That's the annotation is essential for capturing our understanding and our knowledge associated with these things. The genes and proteins are all biomolecules generally. Now, annotation is traditionally, as we said, recorded as free text. You write some text about it. And that's easy for humans to read if it's well-written text, but it has disadvantages, including it's really difficult for computers to parse and know that we're talking about the same thing if it's written by different people. We'll use different terminology, we'll use different terms, we'll write different levels of detail, and that sucks for computers. So these ontologies are a way to use a standard vocabulary to address these kind of problems. So Go, this gene ontology, is integrated with Uniprot and many other databases, including the gene database at NCBI and many others at NCBI. It's got three main parts to its ontology: the so-called biological process. This is a commonly recognized series of events like cell division or mitosis. So think about biological process a bit like it's your cell biology textbook, right? It's where in the cell biology textbook does this gene feature a lot, okay? Then we have a second level, which is molecular functions. This is more like the elemental activity or task or job of this thing. So it could be like a kinase or an insulin binding or a GTPase. This is more like your biochemistry text. Which chapter in your biochemistry text does this thing feature in? And then finally, we have cellular component. And this is really just where the gene product is frequently located. Is it found mostly in the mitochondria, in the inner leaflet, or is it expressed highly in the brain or somewhere else? These kind of levels of detail, which are often very important to knowing what's going on in the function of these things, and particularly important when things go wrong. So back onto the NCBI website here, the gene ontology, or Go, as we've said, is maintained out of the EBI. So the EBI is the European Bioinformatics Institute that we talked about and introduced in a previous video. We're going to actually scroll all the way down right to the bottom to find a link out to Uniprot, one of the major EBI databases and resources here. Now it's all the way to the bottom, so if you just keep scrolling, them pretty much right here at the bottom of the page, because the EBI is still a competitor of the NCBI. But here it is. You see this link here where it says protein accession, and it's got Uniprot. If we click on this, we'll go to Uniprot, because Uniprot has much more detail about protein coding genes, such as this one. So let's click here and we'll end up at Uniprot. This is the Uniprot page. And one of the things that you might notice first is this thing that I've highlighted here in the red rectangle. This is a bit like, like TripAdvisor or some rating site for different restaurants or hotels. It's got this kind of gold star and a rating. This is telling us that this entry in Uniprot is a good one. It's well liked. It's a 5-star hotel. Now, not all of the entries are going to be like this. Some of them will have 1-star, 2-star, and they're maybe not as trustworthy as something like this. This means it's experimentally verified. There's experimental direct evidence and lots of literature to back up what's contained on this page. Now, Uniprot is still kind of unique in offering this kind of rating. I really like it. I find it very useful because not all annotations are trustworthy, as we'll see in one of our projects later in the course. So let's answer some questions here by navigating this Uniprot page. Oh, before we do that, let me point out that one of the key formats here, bioinformatics formats. So if we look at how do we get this sequence here, this is a FASTA format file. You see this sequence? It starts with this little more than or little arrow symbol, some identifier, and then it can be any text on that line. But then immediately after that, the next line is where the sequence starts. and it will continue until either the end of the file, as in this case, or it sees another little more than and another identifier indicating there's another sequence in this file. This is so-called FASTA format, and we'll see this again and again. It's ubiquitous in bioinformatics. It's used all the time, and we're going to get to see it lots and deal with it lots, this FASTA format. All these sequence databases will have an option to get FASTA format files. These are often what we want because most bioinformatics programs will read and do useful stuff with FASTA format files. Okay, so the way we navigate Uniprot here, this is out of Europe, so they drive in the UK on the other side of the road. So here we are on this side to navigate compared to NCBI, and we're going to answer our questions by clicking on the different categories here, which, just like the gene page, will bring you further down or link you to different subsections of this site. So our first question is, what positions in the protein are responsible for binding of GTP? GTP is the guanine triphosphate that this RAS protein hydrolyzes to do its stuff, to turn itself on and off in signal transduction, and we can see that right here at the start. These are the nucleotide binding positions. For example, here it's telling us that positions 10, around amino acid number 10 to 16 or whatever it is here, are responsible for binding nucleotide, as well as some other regions that are distant in sequence. That means they're not directly after one another, but they probably come together when the protein folds up together into its tertiary structure, as we'll see later. So let's bear that in mind. Around positions 10, 11, 12, etc. are responsible for GTP binding. We'll hold that information in the back of our mind as we answer some other questions. Now, another question here that we can answer is, what variants of this enzyme are involved in gastric cancer and other human diseases? Remember the seminar that we were coming out of and trying to dig more background information here out of. So here we see a whole list of different pathology and biotechnology information, including these natural variants. In position 10, for example, is a mutation associated with AML, capillary leukemia, and many other types of Noonan syndrome, developmental disorders, and different cancers for the like. If you scroll down, you'll find gastric cancer and other variants here associated with these diseases. This is very useful information to know. Remember those nucleotide binding positions? They were around 10 too, right? So we're getting some idea, but this is a sensitive site, not only for GTP binding, but if you mutate it, things can go wrong and it can lead to various human diseases, such as gastric cancer, for example. Now, another question we can answer is, if we wanted to go and probe why that mutation site is particularly prevalent in gastric cancers, for example, or why it affects the function of these proteins, we would maybe want to look at the 3D structure, just like we did in our first hands-on session with that beta globin gene and that mutation that was found in that particular patient. So here we could do the same. We could go and look at structure, and this would link to the PDB database. These entries are PDB codes, and we could click on these to open them in a new web browser, such as this one. And so here's our PDB database, the main repository for 3D structures, mostly from X-ray crystallography. And here the question is, can we find where the mutations are located? We can display these files. Here, this is the link in green, or we could do 3D view here. This will show us the format that these things are stored in, the so-called PDB format. I'm just going to click on the 3D view here, then click on interaction. And we can see here's position 12 in the green, right beside this bound nucleotide, right beside the GTP. And these positions 10, 11, 12, etc. are right here cradling where the main chemistry in this enzyme happens, the hydrolysis of that gamma phosphate to yield GDP from the GTP. So we're beginning to understand maybe how these mutations affect function. Maybe they stop or hinder this hydrolysis reaction. That's important for signal transduction in these proteins, as we would have learned from analyzing our results from Go and other databases, for example. Now, if we wanted to go back to Uniprot and find out more about this protein family, its conservation, are those sites that are mutated well conserved in different species, for example, we could link out to other databases here, such as Pfan. Okay, what's known about protein family, its species distribution, number in humans, residue-wise, conservation, etc. Now, Pfan is one of the best protein family databases. It's maintained out of the EBI again, so we click here, off to the EBI, and we can read a kind of Wikipedia-like entry on a given protein family here. We can also click off onto species, and we can see that this kind of chart here showing it's all purple. This means it's all in eukaryotes. We could put our mouse over humans. We could find out that there are how many there are in humans, for example, or any other species here by interacting with this figure. We could also click on alignments here, and we could see all these sequences from different organisms are all lined up. We can see that there's some very common columns that are invariant. They're always the same letter. That means the same amino acid across all these species. These are the conserved amino acids here. We'll learn a lot more about these in the next day, why these are important, why they're kind of functional signatures of these enzymes, and why we want to know about them. Another way to look at this information is the so-called logos here. This is a representation of that same information where the different positions or the different columns in that alignment, but the height of an individual character, how prominent it is, represents how conserved it is. So there you can see positions 10, 11, 12, etc. Those are those glycines, the ones that are mutated in those gastric cancers, for example. Okay, particularly conserved, particularly prominent, and they're present in everything that does this kind of function, this kind of nucleotide binding function, this GTP to GDP kind of interconversion in this case. So with that, I think the video is getting a little bit long. I'm going to stop here and we'll summarize these major NCBI and EBI databases that we've gone through. That's NCBI Gene, EBI Go, EBI Uniprot, the OMAN database, the Pfan database, and finally this protein databank resource for protein structures, and the Pfan database. So with that, I'll finish here, and thank you.